So good morning, everybody. I would like to welcome you to MBI and to my lecture. My name is Grzegorz Rempala. I'm a professor of Biostatistics and Mathematics at OSU. And it so happens that I also, at this time, I'm an interim director of this place. So you guys are actually my guests, for which I am very happy. And I would like to welcome you on behalf of our institute here. It's really great to have you. So today's lecture is an attempt to do two things. And it's always dangerous to do this. So I have to apologize. Before I even get started, it's good to apologize. So I'm a theoretical statistician. And lots of things that I do because I really like the theory that stands behind certain things that are relevant to genomics. So I, at some point, convinced the people who are putting these workshops together that it would be really, really good to at least have a brief review of some of the basic statistical methods that go into some of the algorithms that are being used for differential expression. So, but then I also wanted to make sure that you guys will actually come out of here with the ability to do it. So you would have some ways of actually analyzing gene expressions and be able to identify the ones that are likely to be differentially expressed. So we have divided this course, or this lecture, into two parts. The first part is more theoretical, and I will talk briefly about what is the background and what is the foundation of some of these statistical methods that is needed in order to do the calculations. And then I will have a very, very smart colleague of mine who knows everything about computers, actually has done many of these calculations. He will help with sort of practical aspects and the R parts and the scripts that have been developed to deal with differential expression. There's many different ways in which you can analyze differential expressions, okay? There's no way that I could talk about all of them, so I had to pick and choose. So I actually chose something that I thought was probably reasonably relevant to RNA-seq, and I will talk about a little bit more general and flexible model, which is relevant both for RNA-seq and for microarrays, which are still, even though not actively maybe produced, there is still lots of historic data from microarrays. So I thought that might be also a good reason to mention that. So I will talk right now about something which is called EdgeR, and I will also use this opportunity to tell you a little bit more about sort of an extended notion of regression, which is called generalized linear model. This is sort of in modern statistics an equivalent of what people were thinking about regressions in the 19th and early 20th century. And that has immediate application to this differential expression. So that's one piece. And then I will also talk after lunch about the Lima model, which is the model that is more flexible, and it sort of allows you to do a variety of different types of analyses within the statistical design framework, which was covered by Soledad, I understand, this morning. Okay, so that's the general plan. So let me get started. And again, if there is anything that does not seem to be particularly clear, or if there are things that you would want to comment on, please don't hesitate to tell me. So basically interrupt me at any point. Okay? All right. So differential expressions. So first of all, I would like to talk a little bit about what is a differential expression. So the idea is that there is some amount of activities within the cell that are encoded first in RNA and then in protein, and that amount of activity relative to the possible amount of activity is relatively small. So the underlying assumption in most of the statistical models related to genomics is that there is, at any given time, only a relatively small number of genes or transcripts in general that have been expressed, meaning that they have large abundance. Okay? You can do various types of things trying to identify transcripts which are differentially expressed. One of the obvious things to do would be to just compare transcript to transcript to transcript and see which one of them, say, across different diseased condition, would seem to be enlarged or, in some sense, more abundant than in others. But that's not a very efficient way of dealing with transcripts because, as you can imagine, there's lots of genes and therefore there's lots of transcripts, and we wouldn't be able to really easily do this kind of counting in the types of data and the type of problems that we're interested in. So that's typically not what we do, and that's why statistical methods have been developed to try to make some of these calls for us. So the levels of analyses are sort of going all the way down to the abundance of transcripts. If you imagine, if you're looking at some kind of tissue cellular data, you have ability, again, I'm assuming right now that we, at this stage at least, are not that particularly excited about the details of how this is done, but at some level we have ability to produce copies or some clones of transcripts out of these cells, and we can measure how many of them are there with certain precision. And that's the general idea of how do we count which transcripts tend to be more or less expressed or in more or less abundance in the particular condition. That will tell us maybe something about the dynamics, the molecular dynamics of the particular disease or the particular condition at which we measured those transcripts. So the transcriptome level models are attempts to borrow strength. So these are attempts of sort of looking at overall patterns of all of the transcripts before making a final decision about whether one of them is or is not expressed. Okay, so that's kind of where most of the things that I will be talking about come from, because it turns out that this is often an efficient way or more efficient way of dealing with the notion of differential expression. So we don't want to say that something is differentially expressed just because it's more expressed in one condition than in the other in isolation. We want to see overall pattern of these expressions in these two conditions is such that statistically we can say that these two are differentially expressed, okay? So what I will try to do in the next 35 minutes, 40 minutes or so, I will try to give you just a basic statistical framework that has been developed to sort of deal with this. And then we will try to follow up with some scripting and actually show you how this works in the software that I will be briefly describing. So mostly the analysis that I am interested in is thinking about the data that comes from NGS RNA-seq, but similar type of analysis could be done for other types of molecular data as long as I am really interested in doing some kind of counting. So as soon as I'm counting molecules, these kinds of methods will be appropriate. So differential expression in RNA-seq. As I said already, I have a tag. So a transcript, I will basically call a tag. This very often has to do with the fact that I'm barcoding these different tags, these different transcripts. So in some sense, it really is a molecular or a digital tag that I'm using in order to identify a particular transcript. So I will very often use the notion of a tag to just mean that I'm looking at the particular molecule, or longer or shorter, which is a part of a gene, typically. Okay? So I could be simply looking at the particular tag, and I could be looking at the actual abundance of this tag, like I just wrote over here. I could potentially be looking at something which looks like this, but you see that maybe in these different conditions, it's not entirely clear because of the big biological diversity of some of these samples. It's not entirely clear whether it makes sense to do a direct comparison of these 23 and 42 in some way. So then what people have thought about, for instance, is, well, maybe we can just rank them. To basically build a rankings of these different tags in the sample of all of the tags and sort of see how these rankings compare to each other. At least it would give us some kind of scaling that would maybe be more appropriate and wouldn't be that sensitive to the molecular diversity. So the standard methods that are taking that approach are based on something which is called nonparametric statistics. One of these is called the Wilkinson or sum rank test. The problem with this is that generally these kinds of methods have relatively low power unless you have very, very large number of subjects. So what it means is that unless you have a really large samples of individuals, it is difficult to have good statistical properties for selection of genes in this manner. Even though they are robust in the sense that they have certain ability to be immune to the distortion that's caused, for instance, by the biological diversity of these different signals. So what we would like to do, as I said already, we would like to be able to borrow strength across transcripts in order to boost the power of these differential expression calls. So that's really a fundamental idea. The question is only how exactly do you do it? So there is a number of different ways in which people have tried to do this. The approach that we are using and the software that Maciej will be discussing and some of the examples that we'll show you are based on the so-called EDGE-R or EDGE-R method. And it's one of the very few methods that are actually using the counting as opposed to some other rescaling and some other transformation of your data in order to call differential expressions. Okay? So the way in which this works is basically as follows. So I am assuming that my tag is following a distribution, a counting distribution. So in general, for those of you who have had at least a little bit of statistics, you might remember that Poisson distributions are objects that are very often looked at in the context of counting because they have some good general universal properties that make them appropriate under many conditions. So a very simple model would be, I'm just looking at this count as some kind of Poisson distribution where these Ks would be technical replicates of the particular tag of ith tag in this, and then I would assume that the distribution or the count of this tag really follows the Poisson distribution. And then basically the challenge here would be to identify the intensity of that distribution lambda I, and that would be a way for us to decide whether or not these tags, for instance, when comparing two different tags, if their lambdas would be different, then we would say that they're differentially expressed. So this is relatively okay, and it seems fairly straightforward, and it's generally the statistical idea that almost no one can argue with, except that there is issues with how do you apply this in practice. And the reason for this is there is as you might have already seen in some of the examples that you looked at, there is huge biological diversity that comes in all sorts of these molecular data that we're interrogating. So the life is not as simple as that Poisson model. We really cannot even attempt to pretend that this would be something that would work for us. So we have to make it more complicated just because we have this biological variability that sort of changes potentially these counts and these transcripts in a manner that makes it much more complicated than just calling the intensities or the lambdas of my Poisson distribution. Okay. So here is an example. So if I take a Poisson distribution, it has this amazing property, actually quite interesting, that always mean equals variance. So lambda I, which I called intensity. Actually, one way of thinking about Poisson distribution is that its variance is equal to its mean. So and under certain conditions, this is really the only type of distribution that can have this property. But if I look at the data that comes out of differential expressions, and if I do these tag-by-tag plots, so if I basically attempt to analyze any realistic data, I get something like this. So if I take the logarithm of the mean and logarithm of the variance, I basically see this plot here, which is moving upwards, which certainly doesn't follow the line x equals y. So that basically tells me that my distribution is not really Poisson, that it follows something else because it doesn't satisfy this fundamental property of the Poisson distributions. So that means also that if I stayed on the red line, at least in the vast number of these tags, I would be actually underestimating the variance. The variance is larger. Okay, it's moving upwards. So if I underestimate the variance, that potentially means that I would be calling lots of different things differentially expressed when they are not. So this is the notion of false positive. So if I assume that I have a Poisson model, and hopefully by the end of this lecture, I will be able to show you an example. But if it turns out that if I were to use a Poisson model, I would most of the time end up with horrific number of genes that my statistical method would call differential expressed, but they're in fact not. And that would be something generally very bad because it would make it virtually impossible for me to plan any experimentations or even try to look for anything that might be of interest in the particular comparison of different health conditions. I just would have too many things to look at. Okay? So how does this so I have to basically deal with something which is called an over-dispersion, which means that I have to build a little bit more clever model than the Poisson model that will allow me to deal with the notion that I have the variance which is larger than it would be under the Poisson model. So this is called generally an over-dispersion model. And one simple way in which statisticians deal with this is by means of something which is called a negative binomial model. And it really is the driving model for the rest of this lecture. So the negative binomial model is an attempt to take into account this over-dispersion by basically saying, look, maybe your tag is really coming from the Poisson distribution, but the mean of that Poisson distribution, so here I denote it by lambda, and I'm skipping the subscript. I'm talking about a single tag right now. The mean of that Poisson distribution is not really a constant. It itself comes from some other probability distributions that I will here at least call gamma. This is the distribution which is only for non-negative numbers, and it's this unimodal distribution that can have different shapes. It has these two parameters, R and P. And if I now stick that particular probability distribution into this Poisson model, I end up with something which is called negative binomial. It has two parameters, R and P. There is various ways in which you can think about it. But in some sense, it's a different type of binomial distribution. It's different because it turns out that it can, for those of you who remember what binomial distribution is, binomial distribution basically assumes that I have a number of coin tosses that I am running over a fixed number of trials. So the negative binomial is kind of using a similar idea, except it doesn't have a fixed number of trials. You keep tossing your coin over and over with certain probability up until a certain number of successes happens. And there you stop. So because you don't know when a particular success happens, it basically means that you can have to toss it for a long time. So it basically doesn't have an upper bound. This is called a negative binomial distribution, and it's sort of a blown-up version of a Poisson distribution. So it's useful for estimating counts because it's fairly flexible. It gives you ability to describe lots of different patterns in your data just using this basic concept. So typically, it's parameterized by this thing called R, which is the number of successes that you want to wait for, and P, the probability of a single success. But you can also reparameterize it differently, and in fact, it makes sense for us. One thing before I say that is it then truly, if you basically look at the mean and the variance of the Poisson distribution, you see that it satisfies our condition that we had for it, which is that the expected value or the mean of that distribution should be really smaller than the variance. So it models over dispersion. So I would like to be able to do this in a manner that's a little bit quantitative. So I will reparameterize it by basically taking this new parameter, which now is a function of the number of trials and this basically odds in my tossing experiment. I will call it mu, and then I will calculate the variance. So this is actually the mean, and the variance is here, and I will rewrite it like this, and I will use this notion of mu to rewrite it in this way, when this phi now is kind of the over-dispersion parameter. So if phi would be 0, then I would exactly have that the mean equals variance, and I would basically get a Poisson type model even though, so at least technically I would get a Poisson model. When phi is larger, by the way, phi cannot be negative, so if phi is greater than 0, then I will end up with an over-dispersion. So in order to be able to put it all together, I have to now come up with some kind of general statistical way of dealing with these kinds of distributions. So I hope that I convinced you that Poisson is a good idea, but it really doesn't work for the transcript type data that we're really interested in, that we can try to fix it, at least to some extent, by introducing this negative binomial. But now what do we do? I mean, how do we actually call these different things within the realm of binomial distributions? How do we estimate parameters? So I have to tell you a little bit about this. Okay? This is really the main idea that was used by the people who developed the Edger or Edge-R software. And I will talk about it briefly. So this is a digression. This is actually an opportunity for me to tell you in a couple words, 2 slides, I think, maybe 3, about something which is called the generalized linear model. So in statistics, nowadays, if you're looking virtually at any problem that doesn't have to do with statistical learning and you know, which can be described by some kind of regression type relation, you are no longer looking at traditional linear regressions, but you're looking at something which is called the generalized linear model. So generalized linear model is an attempt to generalize the notion of regression to nonlinear relations. Very heavily nonlinear relations, not even necessarily polynomial. Very, very strange types of relations that will mix together both continuous and discrete data. In many ways, this is what we are after, because we will have discrete responses. These are these different transcripts that we would like to measure and decide which ones of them are differential expressed. But we may have lots of additional data, information that tells us where these transcripts are coming from. So we may have various information about the patients, about the different tissues, about different disease conditions. So we would like to be able to build a flexible model that will allow us to use all of that information to potentially help us with making decisions about what should or should not be called differential expressed. So we need a general way of analyzing this generalized linear models. Okay? So very briefly, I will tell you what these things are, and hopefully it will make it easier to understand where we're going with this. So the idea of a GLM is that I have a very special type of distribution. So this distribution, it's called an exponential family. It turns out that lots of distributions that we see in real life, maybe it's not immediately obvious, but many of the discrete ones, so for instance, Poisson distribution, as well as a binomial distribution, as well as a normal distribution and gamma distribution, they all can be written in the form of this exponential family or exponential dispersion family. So the idea is that the density of a function like this, or a probability mass function, can be expressed in terms of an exponential function, which is written in this form, that you have some parameter which is multiplied actually by the observed variable, minus some function that only depends on the parameter. It doesn't have any variable in it. This is under the exponent, and this is divided by some parameter which is now a scaling parameter. It's in the denominator. So it's very important that you have this structure of y times theta, where theta is unknown parameter, potentially unknown, minus something which is only a function of that parameter, divided by something which does not depend neither on y nor or theta. It has some parameter phi that we basically call dispersion. And then on top of this, you basically have maybe some additional factor which only involves y's and phi's, but it doesn't involve a theta anymore. If you have a structure of this type, it turns out that you are looking at something which is called an exponential dispersion family. Very often this a of phi function is just phi, or it's just phi divided by some weight, wi, which is known. Phi is called the dispersion parameter because in some sense it affects the scaling of the argument of these exponents. So this is sort of a way in which we are scaling things up or scaling things down. Now this piece over here, it's like, because it's under the exponent, it's a multiplicative factor. So in some sense, it's just something that sits to the side, and it doesn't affect, other than by multiplicative scaling, the shape of this density function. So now, it turns out that if I have families of functions like this, there are some nice properties that they have. For instance, the expected value of these yi is just obtained simply by differentiating this b function here. So if I know what the b function is, take a derivative with respect to the parameter, then it turns out that this is nothing else but the mean. And in a similar fashion, if I take the second derivative of b and multiply it by my dispersion, then I end up with the variance. So these are just some properties of these functions which are very helpful in some sense. The exponential dispersion family is the nicest possible family in statistics of distribution functions. About these families, we know almost everything that we need to know. They are wonderful. They're simple. They're easy to understand. We know what is the data compression. So why do we care at all? Mostly because we would like to understand where does the information about certain unknown factors, in this case, for instance, about parameters, comes from. So it turns out that in these dispersion families, we have a very, very good idea about what is the amount of information that we need to have in order to be able to analyze the parameters. These are called the so-called complete sufficient statistics. And really, in general, these are the only types of statistics or families for which we know this. So, for instance, as I will tell you in a moment, it turns out the negative binomial distribution actually is one of those that are describable in terms of GLMs. That's wonderful because all of a sudden, as you will see, we have a relatively straightforward way of dealing with calling some of its parameters, and that will lead to an algorithm for differential expression. Okay, so moving forward, Poisson distribution, as I said earlier, is a special case of this exponential dispersion family and can be written in this form just by algebraic manipulation. And then you're looking at something which looks like an exponent. Here I have this yi times log of lambda I minus lambda I. The dispersion is not here, so that means that I will take a of phi to be 1. There's just no dispersion parameter here. And then I have this additional piece on this side. So it basically looks like GLM with these parameters. And sure enough, if I take a derivative of this function b, which I basically wrote here, with respect to theta, for those of you who remember your calculus, if you take exponential function and differentiate it with respect to the argument, it's just the same function. So it means that if I differentiate twice my b function, I end up with e to the theta, but this was lambda, so it basically says that both the mean and the variance of the Poisson distribution are lambdas. And I can play these games, with other examples. This is a normal distribution, so I don't really want to spend too much time on this. But in the similar fashion in which I have dissected Poisson here, I can also dissect a normal distribution and also obtain some formulas for the mean and the variance and some other things that would be of interest in the future. So negative binomial is another example of this dispersion family. So if I write my negative binomial in this form, that this is negative binomial with these two parameters, then the inverse of R, I will call over dispersion parameter, I will call it phi. That will be consistent with the parameterization that I just shown you on the other slide. And I can rewrite this formula in terms of this exponential family. So if I do this, I basically see immediately that my theta I, so this parameter, this is, by the way, often called the natural parameter in this setting. So this parameter theta will just be a function of mu and r. My dispersion is unity, so there is no additional dispersion here. And I can calculate my function b. So with all of this, it seems that in order for me to be able to say anything about my parameters, I have to figure out a way, how do I estimate these kinds of quantities, which are mixing the mu's and the over dispersion parameter R. Okay, so that's the goal. So here's the package. So the package that was developed to deal with this. So there is a straightforward method of estimating parameters. It's numerical, because most of the time we cannot write out the exact solutions of the appropriate equations for estimating parameters. But we have a very nice, stable numerical method to do this as soon as we are within this dispersion family. So EdgeR model was developed with this in mind, that we have differential expressions, which are describable by negative binomial distribution. And we would like to be able to do some flexible analyses of those differential expressions from the cDNA libraries. So this is the Robinson and a couple other people who were involved in this. This package was created in Bioconductor a couple of years ago, sort of at the dawn of the RNA-seq methodology. So the idea is that for tag I would really assume that I no longer look at the Poisson distribution, but instead I look at the negative binomial distribution, which I have reparameterized with these two parameters, mu and phi. But now, for mu, I will basically have lots of flexibility. And typically the way in which I would really, so the mu is this mean that I was talking about earlier, but it's also a way in which I can relate, like in the regular regression, a potential covariance and parameters to my distribution. So one way in which people are doing this within this general notion of GLM, and I didn't say that, and maybe I should have earlier. So the GLM basically consists of two pieces. There is what we call a piece which is stochastic or random, and there is also a deterministic relation. And this is basically where the regression comes in. So there's a relation between this parameter that I have created sort of artificially over here and the original mean of the distribution. And through this, you have ability, if you parameterize this mean in some way, you have a way of basically parameterizing your unknown parameter by means of things that are of interest to you. So for instance, in the regression model, we would use coefficients on the covariance. So this is an example where this is done. So I would basically take this G inverse, and I would take it on some kind of regression model when I have a bunch of covariates for my tag, and I am multiplying these covariates by the set of coefficients. And then these x's, which are my covariates, this is something that I would know, this would be given to me, and I would be interested in estimating betas. So basically, I am converting this entire problem now from looking at the means to looking at the coefficients that are sitting by my covariates, okay? And I can use exactly the same methods and the general machinery of these generalized linear models in order to be able to estimate what I need to estimate in terms of the beta coefficients. So that's sort of a standard trick that people are using, and that's one of the reasons for introducing these generalized linear models in the first place. So assuming for now, just to make things a little bit simpler for me in explaining it, that I don't have any covariates, so the means are just the means, I can now calculate, because I am in this family, which is a GLM, and it's the dispersion family, so I can, exponential dispersion family, I can basically identify what is the least amount of information that I need in order to able to do estimates of my parameters. And the least amount of information is actually, as it turns out, and that comes straight out of the theorem that's in statistics in this setting, it's just a summation of all of the counts for that particular tag across all of the subjects. If I have the sums of that particular tag across all of my subjects, this is all the information that I need in order to be able to estimate my beta coefficients. There's no more information or no less information that I can collect in some sense here. So this is the so-called minimal sufficient statistics, and it's also complete. It means that it has really, it's the maximal compression of the amount of information that you can, in some sense, provide in this setting. So in order for me to be able to say something about these coefficients beta, or which is equivalent about mu, all I need to do, I need to look at all counts possible, okay? For that particular tag. So this is an example. Again, Maciej will talk about it in more detail. We looked at the data from the breast cancers, and we have analyzed a number of subjects, I think 15, when we looked at different stages of breast cancers, and we looked at the RNA-seq data that was available to us. We looked at these after normalizations and after adjusting for the library sizes. Maciej will talk about it in a moment. But this is basically how the data, the original raw data, looks like when I am downloading it from the summaries of the RNA-seq count. So I'm basically looking literally at counts of different transcripts of genes, or I call them tags, across different people, okay? So now it turns out that in order for me to be able to say something meaningful about the parameterized version of this problem, I really need to sum these counts up and look at them in some way, okay? So this is the Z statistics that I was talking about. So these are all tags in my data. I have to do two things. So what I told you is exactly right, except that not quite. So the one thing that I really didn't discuss is this parameter phi, which I have here. It's a nuisance parameter, meaning that I really don't want to deal with it, because in some sense, that's not the interesting parameter that I want. This is just a scaling. But this scaling obviously will affect my ability to tell whether the mu's are the same or not. If I get it wrong, I might see that there is a larger difference than there really is, or vice versa. So I need to take care of the phi. So the first thing that this EdgeR software does in the context of the GLM really is it attempts to estimate how does the phi look like. So the scaling is really key. As you will see in a moment, it really is. So in order for us to do some kind of scaling, we need to calculate this overall dispersion factor. So the overall dispersion factor is really a dispersion factor that is looked at across all of the tags. What I was talking about up until now is tag specific, meaning that I am running this negative binomial model on a specific single tag. So I have up to 20, 30,000 different binomial models sitting in the data set. But now I'm talking about something else. I'm saying I would like to calculate overall dispersion. What does it mean? Basically means that I am looking at all of these different models for single tags, and I'm assuming, for a moment at least, that they all have the same scaling. It's probably not true, and in fact, most of the time, this would be a bad assumption in itself, but I will use it at least to make certain things look a little bit cleaner. So I will obtain this overall dispersion parameter, and that, again, can be done using this model that was developed in EdgeR. And then what I will do, I will develop a model in which I will try to take what I have calculated from this overall dispersion parameter, and I will try to relate it to what I have calculated as my dispersion parameter for a single tag, which basically means that I will be basically mixing two things together. I will be looking at the overall dispersion parameter, but at the same time, I also will be looking at the single dispersion parameter for a single tag. And I will be mixing these things at some proportions. I called it alpha and 1 minus alpha. So that means that I'm taking a little bit of this overall dispersion, and then I am taking the rest somehow from the dispersion of the particular tag. And I will then use this one as a single estimate. This is called the shrinkage method. And what it does, basically, it allows you to, in some sense, decrease artificially the noise that comes out of estimating these separate tags one by one. So the effect of this is that if this is my phi, which was basically the overall dispersion that I've estimated from the entire model using all of the tags, and if these dots are basically the estimates of the over dispersion parameters from a single tag, then what I am doing by basically doing this smoothing that I have shown you here, I am basically running the shrinkage line. So I'm basically taking, and by taking the bandwidth and calculating these things on average, I'm basically running a sort of regression on these. Whenever I get too close to the red line, I will just make it equal. So I will basically then, I will try to shrink down these points here to the red line, towards the red line. What does it do? Well, it makes for a pretty picture. That's the only reason. No, it basically turns out that it actually improves our ability to identify differences in these patterns. So basically, it cleans up the pattern to some extent. So this is an example when I have used a number of subjects and 5 repeated studies. I had the 5 people and the 40 people. And I have looked at 2 different ways in which I would estimate my parameters that I will get to in a moment, the betas in these studies. The dotted lines are the ones in which I use the shrinkage idea for estimating the over dispersion, and the solid ones are when I did not. So what you should be able to see, that I had a much, much larger variance overall. And also, the reproducibility of this data set is lower than if I actually use the shrinkage over here. Okay, so reproducibility, meaning in all of these 5 studies, I was much, much closer to the distributions of these parameters were much closer to each other. And then if I have 40 people, the difference is not so much because I have much, much more data. So generally, the shrinkage method doesn't seem to be make that much of a difference. But on the other hand, it still makes things slightly better. So in a way, I could say that I don't need to use shrinkage if I have lots of data. But on the other hand, this seems to be indicating that it really doesn't matter. I can use it because it's not going to hurt me. And in fact, in most cases, it's going to help me. It definitely helps me when I have very few data, which means that these over dispersion factors are large. Okay, so there's a function that works within the package that we'll be looking at in a moment, which is called estimate common dispersion. So that's how that basically runs an algorithm, numerical algorithm that will calculate this phi, and then it will estimate the common dispersion, and that common dispersions can be then used to do another estimation, which is estimate tagwise dispersion, that will allow you to calculate these shrinkage factors for the appropriate scaling. And that's really crucial. It basically makes the entire analysis go on a much more solid footing because we have less possibilities for making large errors just by misscaling or by misdividing your actual data at the end of the day by some factor, okay? So if you guys remember, if I have a small factor in the denominator, I can make the number inadvertently very, very large. So that's always a problem with these kinds of comparisons when I'm using some kind of scaling. So the tagwise analyses, in the tagwise analysis, I will be scaling ultimately these counts that I am collecting. So I would like to make sure that I use shrinkage. So we'll talk about it in a moment. So then how does the differential expression works now that I have the idea of how do I deal with the over dispersion? So let's say, for the sake of simplicity, I am comparing, so it's a special case of what I was talking about earlier. let's say that I would have a number of these different data points from different individuals, different subjects, in our case, 15 individuals with different stages of breast cancer. And I would basically code it in such a way that I would have these covariate X, which would say you are either in group 1 or you are not. If you're not in group 1, you get a 0. If you are in group 1, you get a 1. That's basically what this writing means. This is called an indicator function. So it's a 1 whenever the statement in the brackets is true, and it's 0 otherwise. So if I do this, I've basically created here a little regression, which is really running on these integer values, which says if you're in group 1, then your value is beta 0. If you are in group 2, your value is beta 0 plus beta 1. So then beta 1 basically has this interpretation of the difference on average. Not really on average, because I'm running this thing through some fancy function here. But if G is, for instance, identity, then it is really an average between these two different groups. Okay? So the idea is that I can have this parameter that somehow is representing the difference between two different groups. What I would like to do, I would like to be able to use this now to say which of the genes in my analyses across these two different groups are going to tell me the most about this parameter. Okay? That's how I'm going to identify the differential expression. Okay, so the way in which this is done is I'm basically looking at these partial sums, Z1 and Z2. They're calculated on the basis of one group and the other group, so they are both negative binomials, according to what I just told you earlier. And then I am doing the usual thing that statisticians do. I will make an assumption. I will test the hypothesis. So in order to identify things which are differentially expressed, I will make an assumption that things are the same across these two conditions. And then basically, in my language now, it means that I take beta 1 to be 0. If I take beta 1 to be 0, then it means that all of these guys have this adjusted mean to be beta 0, or beta null. Okay? So if I do this, then what's the logic here? I would like to find out which of the genes, or these tags that I have created now across these two conditions, will be most in violation of that hypothesis. Okay? So the way that this is done, I create this p-value for a particular tag. So how exactly is this done? So it's done as follows. I am basically looking under the null hypothesis that's H0 beta 1 is 0, I exactly know what are the distributions of the Z1 and Z2 for each tag. So I can basically calculate what you guys might know as a p-value. P-value is basically the probability that under the particular hypothesis you will see the value of your statistic or your data, which is as big as something or larger. Right? So this is basically the tail of your distribution. So I can compute this p-value, which is here. This is under the observed. And then, this is, after all, a discrete sum. I am basically counting things in Z1 and in Z2. So I basically can calculate this overall p-value based on how many times or how many different values of Z1 and Z2 do I have that will basically generate me a p-value that is smaller, so more extreme, than the observed one. And then I will normalize it, because this number has to be between 0 and 1. So it basically means that I am taking this on the line, Z1 plus Z2 is equal to the observed value. So, right, so this is this Z observed is Z1 plus Z2. And then I'm taking all possible ways in which I can split up these counts in the first and in the second group in such a way that my p-value is more extreme than the one that I have observed. So it's basically a way of defining a new p-value based on these two observed counts, Z1 and Z2. Okay? And then I will be looking for each tag, I will compute this p-value. And then, at least at this level, before we get to more interesting things later on, this will be my criteria for identifying which of these tags will really fail, meaning that they will have a tiny p-value. So under this null hypothesis that there is no difference between these two groups, they will actually be very unlikely. Okay? So that's the general idea of how do we deal with this. So that's the p-value computation, and this is called an exact test. Okay, so this actually is a calculation which uses this straight out-of-the-box GLM idea that you can use negative binomial counts. It works, but sometimes if you have lots of data, and for instance, very large data sets, there is a faster way of computing these p-values. And it's based on another idea, which is called the likelihood ratio test. And again, it uses the similar notion that I am assuming that one of my parameters, beta 1, is 0. And then I am looking at the log likelihood, again, under the log likelihood, which basically means at the probability distributions of these negative binomials. And I will use the fact instead that I can combine these p-values in this fashion. I will actually use the fact that if I take the difference of the logs of these things, they kind of look like a chi-square distribution. So it's another statistical distribution that I can use to approximate these ratios, and then I can calculate p-value based on this chi-square. So then it's called a non-exact test. It's called the chi-square asymptotic p-value. Okay? Both of these are calculated as part of the analysis when you're looking at the EdgeR software. So here's an example of the EdgeR at work. Okay? This is ultimately what we would be interested in doing. This is the data which is looking at the concentration versus fold change in stage 2 and stage 3 and stage 1. So we actually took the two most extreme in the data set conditions, stage 3 cancer versus stage 1 cancer, and we wanted to look at the genes that tend or seem to be statistically significantly differentially expressed across these two conditions. We don't care which direction. It's either or. So if I run a Poisson distribution, remember I told you earlier, Poisson, not particularly good because of this over-dispersion business, oversimplistic. If I try to just straight run Poisson distribution with my negative binomials, not caring about the these tagwise over dispersions, I basically get everything red. Everything lightens up in this analysis. All of my genes, or almost all of my genes, are all of a sudden differentially expressed. That doesn't make any sense, and generally this is, I am skeptical that that even has any chance of being true. If that would happen, we would fall apart, by the time we would be born, we would be no longer a feasible biological system. Biological system has to be able to be homeostatic, meaning that it has to have the ability to control its signals. And this basically tells you everything is a signal. That's crazy. So that doesn't work, and that's one example of why these models cannot be overly simplistic, because then they are just not very useful. So if you do a Poisson model here, if you just look at the Poisson model, it's bad. But now, if you look at these tagwise model that I have just shown you, if you basically look at the p-values, then at some reasonable level that we'll discuss after lunch, you see that I get much better general notion of what is differential expressed. So all of a sudden, these red dots are relatively few, and they're sort of in the places when you would expect them to see, in the extremes of these log plots. So that's kind of the general idea. Now, what we'll try to do, we will try to show you how to actually run this analysis. So now that I have shown you how this works, you hopefully by now have a basic notion of what this EdgeR does. It is a model that uses over-dispersion as one of the tools in order to identify these differentially expressed genes. First, you have to calculate over-dispersion. Then you can use either the exact or approximate test to calculate the p-value, and then you rank by the p-values your different features, in this case, genes. Okay? So that's how it goes. And I have shown you just a couple of these screenshots. We have a script that we'll actually try to run, and Maciej will explain how this goes. So we basically tried to give you a little bit of a walkthrough that's following the logic of the lecture up until now, so you guys can actually try it. Hopefully at the end, you would end up with producing something which looks like this, at least closed. Okay?
Yes, so now you got a pretty good background, and right now we will try together to kind of run the analysis, and you will be identifying genes differentially expressed between two groups. So this, we will start with downloading differential expression folder from the box. And in the box, you got two modules. In this folder, you got two modules, Edger and Lima, that we will use after the lunch. And you would have like two files. One Edger assignment. So basically, this is brief description what I would like you to kind of try to figure out how to do based on the presentation and with the support of Edger code. That should contain all the necessary steps to run the analysis. And you got also Edger data. And so this is CSV file that you will read in the code and analyze. Yes. So right now, let me briefly walk you through the process. And then we will. Then you can start playing with the code, and you can start analysis. And I will be walking around the room and helping you. So basically, we will start. If you haven't installed yet, installation of Edger package. So as you remember from yesterday, Edger is located in Bioconductor. So first you have to call this Bioconductor source, and then you use bioclite command to install Edger, but again, this should be already installed, yes? And to start the analysis, you have to load library Edger by typing library Edger. And then you set path to the working directory. If you click the code that is provided in the folder, you should start directly in the working directory that contains the dataset. And you will load the dataset using command read CSV. That is pretty similar to the command read table that reads any table into the R object. And then, as you will see, you would have like all table, and in the first row you would have the sample names, and in the first column you would have gene names. So then to format it into the table and to the data objects that would be processed later, you have to if you open with Excel Edger, you will see the first row would contain the tags. If you read it using read CSV, this first row would become the column names of the R data object. And then you have to move first column and make row names using data included in the first column and remove it. And after processing it, hopefully when you type head counts, that would display first 6 rows of the R object, you should see something like this. And later we have to define groups. So right now, as Greg mentioned, we would compare stage 1 with stage 3 of breast cancer. And we will have like 5 cases in each group. And second step would be building Edger object. So using DGE list command, providing information about read count table. So the table with the read counts would be put together with the group information that would tell the model basically which group is stage 1 and which is stage 3. So, and one more word about like Edger object. So this is like R-specific data objects that contain all the information that the subsequent function need for the analysis of the data set. So you can consider it as a sort of a folder in which you put sheets with the count information, the stage information, and all the other information you need. And whenever you do any computation, for example, when you compute common dispersion or tag-wise dispersion, it's also put into this folder. Yes, and this is this CDS object that you will create at this point. And when you type names CDS, you will see what kind of tables or what kind of sub-objects you got in the CDS object. And again, you can call them and see them using, for example, the head function. So head CDS counts would display the count table, and CDS samples would display the sample table. Yes, so this is the first thing. And here you can check how many samples and how many contain genes with 0 counts, and in this case should be 0. And this is when you type CDS, this is basically the kind of brief overview of the data included in this object. And here you can do some quick pre-filtering. So basically you would remove the genes with very low signals that might affect the analysis. And as you will see, after checking the size, you should have a couple of tags removed. And then you calculate normalizing factors for these samples. And you will see that the norm factors would be adjusted. So here was 1. So you had each sample was considered as 1. Here is adjusted. And you can also check library size by multiplying the library size by normalizing factor. And the next thing you would be asked to do is multi-dimensional scaling plot. So basically, in this plot, you will see overall similarity of expression profiles. Here would be on the basis of the top 500 most variable genes. So basically, you take these top variable genes and you see how samples or how the expression profiles of these samples are similar to each other, or whether they group, whether they create any clusters and such. So this would be, next step would be mean variance plot. So you can compare the Poisson assumption with your actual data set. That you should see something like this. So this is this Poisson line, and this is your this dispersions of your actual samples that you will be analyzing. And right now, differential gene expression. So as Greg mentioned, you would use exact test. And to do it, you will use function exact test. And we will do some small experiments. So basically, you will run exact test with common dispersion and with tag-wise dispersion and Poisson. And you will be able to compare and see what happens and what are the values, basically. Yes? And for each object, the output object of exact test function processing, you can see basically what are the items within this object and what was the comparison. So basically here you got this group names that you can call by calling the comparison, basically. And you can see what you got by typing the name of the object and the table where you would have fault change, log counts per million, and p-values for each analyzed tag. To get genes differentially expressed with like a false discovery rate correction, you can use function top tag. And in the function, you provide the data object, the results of exact test analysis, with the number of rows you would like to display. And you tell basically how you would like to sort, whether by a p-value or like fault change. So here are the options. In this example, you will change the fault p-value, I'm sorry. And this is what you should see. And if you would like to see whether this like FDRs and p-value and fault change is based on some kind of outliers that affect the results, or whether it's consistent, you can see the row count table with the order of the genes by p-value by typing the CDS count. So basically, you call the object counts from CDS data object with the ordering of the row names from the table, from the result table. And then you can save, or like you can assign the values to the results for like common tagwise and Poisson models using a top tag command again with the appropriate like result file. And as an n instead of 5, you put a number of rows of the actual data object, yes? And for these genes, you can also select genes that were significant, and you can define significance. In this case, it's p-value, adjusted p-value 0.005. And you can see how many genes were differentially expressed. And you can also like express it as a percentage of genes differentially expressed. And if you'd like to see which genes were up and down regulated, just kind of a general summary of the statistic, you use the side test with providing the result data object and define the criteria on which you will decide whether it's differentially expressed gene or not. And this is what you should see with genes down, up-regulated, and no change. And yes?
So you have three things you've got on there. It's the POI is the Poisson, and then the TGW is the
tagwise. Common dispersion. So use the first examples that, I mean, second after Poisson that Greg was presenting. Mm-hmm. And here is example how to generate MA plot. So basically, we will use a plot smear function from Edger package. And this is the result that you should see, basically, after running this command. And here, just a couple of words about the outputting the results. So basically, you would like to see the list of genes differentially expressed and fold change p-value based on which you can decide whether genes are differentially expressed, plus the read count table, so you can eyeball and you can just confirm that these differences are consistent and are not the effect of some outliers presence in one or another subgroup. So basically, here we can adjust the columns by matching the row names of the output tables with count tables. That will happen here. And combine the results using a function cbind that basically binds the columns of R objects. And here we would bind together first the result table and the information about tagwise dispersion for each gene, and the information if it was up, down regulated, plus read count table at the end, all sorted by the result row names that would give you basically the genes sorted by the p-value or FDR in this case. And this is what you should see. This is the result. And the same you can do for all the other tables. And writing to the disk. So after having all this like analysis done, you would like to save it, yes? In this case, we would save it as a CSV using function read table. You can use also write table. You can also use the function write CSV, where you write your huge file with all the information you need. Provide a file name. In this case, you provide separator comma, because it will be a comma separated value file. And that's it. So right now, you can start with this like brief PDF that would have several steps. And if you can figure out it by yourself, please do it using the presentation one or another presentation. If you got problems, you can always go to the Edger code And you can write the code. You can run the code and get stuff done, yes? And you can also run it to check to make sure that your results are similar to what we showed.